I would, yes, thank you very much. Good morning. I would like to start uh, today's class. Today we shall discuss on a special topic, not uh, uh, phenolics, terpenoids, or alkaloids. We shall discuss on the analytical methods, uh, analytical processes or analytical methods which have been used in discovery of new bioactive natural products. And already you know what are natural products and what is chemistry and natural product chemistry, all these things I am not going in detail. This is the main slide you must follow carefully. You know that bioactive natural products, especially we discover from the plants, microorganisms, marine organisms, or animals, and all the source organisms, they biosynthesize the compounds in their cells, and scientists discover those compounds by analytical uh, methods, following analytical methods or analytical processes. For example, this is a plant, and plant bark contains bioactive natural products. So you can take the bark, plant bark. So it can be, in case of other organisms, for example, fungi or bacteria, the whole cell of the organism, or other organisms, even insect, you can take the whole organism, whole insects or like that. But for any analytical process, you need sufficient amount of you need a sufficient amount of materials, because uh, most of the bioactive natural products present in plants or other sources in a very low concentration. This is why we need enough amount of materials for the analytical process to get a sufficient amount of or detectable amount of bioactive natural products. So, for example, if you take bark of a plant, for example, then what you need to do to dry the bark in shade and not in the light condition or sunlight, because sunlight, some compounds are sensitive to sunlight, so sunlight can cause decomposition or breakdown of some of the bioactive compounds. So some compounds are photoactive. Photoactive means in presence of light, they may cause breakdown, and you can lose the targeted bioactive natural products. So uh, after drying in the shade, you need to grind and extract with solvent. Yes, what is grinding? By using a mill of grinding machine, you can make the bark into powder. Why? Because bioactive natural products present in the cells. So if you crush the material and make them powder, then it would be easy for the solvent to get the bioactive compounds or chemical compounds. So after grinding, you will get the powder, and this powder you place in a flask, conical flask or some big bottle, glass bottle, and add organic solvents. Organic solvents for extraction. Extraction means to take out the chemical constituents from the natural materials like powder of the bark uh, into the solvent. Solvent in case of extraction, we use different organic solvents, for example, ethanol, uh, methanol, or acetone, or ethyl acetate. Several organic solvent we can use, but usually in case of bioactive natural products in small scale laboratory assay, we use ethyl acetate or acetone because we use ethyl acetate or acetone, and we can get after you may shake the flask occasionally so that all the cells of the plant material gets the solvent. And three times, usually we use three times. So take out the solvent by using a filter, and the residue again you extract with the same solvent. Sometimes solvent also contains, we use water, for example, 40% water containing acetone, or 40% water containing methanol or ethanol, like that. Anyway, when you get the filtered material that is organic solvent, that organic solvent contains the uh, chemical substances, that means extracted the chemical substances and present in the organic solvent. And organic solvent, then you evaporate the organic solvent by using vacuum evaporator. And after you will get gummy like dried material that contains your compounds, bioactive target bioactive compounds with mixture of other huge number of compounds. In that case, you need to use some separating techniques, analytical techniques like TLC or GC or HPLC. TLC means thin layer chromatography, GC means gas chromatography, and HPLC means high pressure liquid chromatography. These are the analytical techniques used for separation of the compounds from the mixture. So, for example, this is a TLC. If you drop a microliter of the ethyl acetate solution of the extract, and if you run in a TLC tank, the materials will move upward, and here you can see several spots. That means different compounds gave different spots, and you can understand how many compounds and understand the number of compounds and present in the mixture, extract, as well as you can understand the polarity of the compound, or like that. HPLC is another technique. We can use the crude extract, and usually semi-purified extract we use for purification of the compound by using HPLC. These techniques are available in our central laboratory. You may, when university will be open, you can see and visit and see there. These techniques are used to separate HPLC. You can get the purified compound. If three or four compounds are present in the semi-purified extract by using some other chromatographic technique, then we can use the HPLC because it is very expensive, and we can only separate the or purify the compound in small scale. So preparative column chromatography this is the preparative column chromatography is very important in the laboratory. This is a glass column, and filled with the silica gel or other adsorbent. and you use a small amount of solvent to dissolve the extract and put here, and then you add a certain ratio of the solvent, for example methanol and ethyl acetate, um, for example 1 is to 99 or 5 is to 95% or like that, and then the compounds pass through this column. When pass through, depending on their molecular weight as well as polarity, they will move in different speed and separate, and you can collect in a test tube, in the test tube, so for example 10 ml, 10 ml, 10 ml. So and after that, if you do the TLC, you can find highly separated compound, and sometimes you can find the pure compound also. So after purification, only one column chromatography do not allow you to purify the whole mixture of the compound, and after getting different fractions, you can test where your active compound is present. How can you do it? By using bioassay. You can test each fraction which one is bioactive. For example, if you want to target uh, a compound which is inhibitory against the wheat blast fungus, then you test each of the test tube containing compounds in the against the wheat blast, and if you find the fraction 5 contains the wheat blast inhibitory compound, then others no need to concentrate. You only concentrate fraction 5, and then if you confirm that it is wheat blast inhibitory compound, that is fungicidal compound, then you go for different kinds of analytical techniques called spectroscopic method. Spectroscopic methods to elucidate the structure. What is the chemical structure of that fraction? So we use usually mass spectroscopy and NMR, nuclear magnetic resonance, and some other spectroscopic technique like CD to determine the chemical structure of the compound, and then structure is elucidated. If the compound is new, not yet been discovered from any sources, in that case, it is it is sometimes needed to synthesize this compound in the laboratory and see whether synthetic compound and the natural one have the identical spectroscopic data as well as the biological activity. Once the biological activity as well as the spectroscopic data are same and identical, then your structure is confirmed. So elucidation of chemical structure is a rigorous process and called spectroscopic methods. And spectroscopic methods, there are many types of spectroscopic method to elucidate the structure. Here, analytical methods, especially the spectroscopic method, for example, mass spectrometry. Mass spectrometry we use to determine the molecular formula as well as the molecular weight of a natural product. There are several types of mass spectrometry, for example, Fourier desorption mass spectrometry, electron ionization mass spectrometry, high resolution mass spectrometry, multi-TOF mass spectrometry, that means metric assisted laser desorption, time of flight, ionization time of flight mass spectrometry, that is metric assisted laser desorption ionization, time of flight mass spectroscopy or spectrometry. LC liquid chromatography mass spectrometry. This is another HPLC mass spectrometry. Sometimes we use HPLC high pressure liquid chromatography connected to the mass spectrometry. So after purification of the compound, immediately we can get the mass data, that is molecular weight or molecular formula. There are others, I told you, spectroscopic method, NMR. Nuclear magnetic resonance can be carbon NMR, proton NMR, and two-dimensional NMR. NMR is used to determine and to understand the nature of carbon and proton. Proton means hydrogen present in the chemical structure. And they give nice peaks, and from the peak we can determine the whether CH2 or CH, how many CH2, how many CO, what kind of carbons are present in the structure. And proton NMR gave the information of all protons. Protons means hydrogens. Hydrogen can be present in a structure in methyl form or hydroxyl form or methane form or any other form. So uh, proton NMR gave the all proton information. How protons and carbons are interacted, two-dimensional NMR will give us the information based on this mass data, proton NMR data. We can determine or infrared, spectroscopy, IR or CD, UV spectroscopy, comparing all the, considering all the spectroscopic data, we can determine the structure of a molecule. And once you determine the structure, and if it is a new compound, it is a new discovery, like, the penicillin was discovered by Alexander Fleming. So natural product chemistry is very, very exciting for discovering new drug candidate or new candidate for the agrochemicals or antimicrobial substances or new molecules for hormonal and other biological activity. And in industrially, it is very important, and usually small scale analysis is done in the laboratory. Once you discover the compound and know the properties of the compound, then the big scale industrial process is done by the industry. But in the university laboratory, small scale analysis following these techniques are used. First chromatography for separating and purification of the compound, and after purification, spectroscopic methods are used to determine the structure. You must understand that if compound is not pure, uh, uh, then you cannot get any benefit from these using different spectroscopic chromatographic method, and then go for the spectroscopic analysis. Thank you very much. This is a short lecture for today. So what you need, you can we can discuss more about the lecture, and you can ask the question. So it is now open for your question, and we can discuss more if you ask any question.
Assalamu alaikum, sir.
Walaikum salam, Muaz.
Muaz, sir. Sir, I have a question regarding your lecture about the analytical process of discovering new compounds. Sir, if you go for a new compound and we do all of the procedures, but finally we get chemicals that is already been discovered. So what, so what is the end of this process? The process is in vain or something, sir? We didn't get anything, sir.
Yes, in case of bioassay guided, fractionation, that is chromatography and spectroscopic methods to elucidate the structure. If you get a compound is known, already been discovered by other people from other sources, uh, your question is whether the result has any meaning or not. Yes, it has meaning if a new biological activity of your known compound you can establish. For example, if you find your compound is active in inhibiting or suppressing the wheat blast fungus, which has never been tested by any other researchers, then you can, this is a novel and new findings, new discovery of a known compound. So you can publish it and it is a very good value. It has a very good value. On the other hand, if your compound is was discovered from microorganism but you discovered from a plant source, a native plant source, then it is a new source, at least some value. And if it is discovered from the same source and same biological activity, nothing new, then it is not interesting and you cannot publish the data. But usually bioassay guided methods result at least new biological activity. But in natural product chemistry, sometimes scientists, after discovering a compound, if they find it a known one, they becomes puzzled. And most of the cases it is known one, because scientists over last 1 century, after the discovery of NMR and mass spectroscopic method, uh, almost touched all the plants and even most of the microorganisms, most of the important classes of microorganisms. So this is why to discover a new compound is really challenging. But even though every day scientists all over the world, they are discovering new compounds from natural sources. So natural product chemistry is a fascinating subject. Like Sir Alexander Fleming, uh, once you discover a new bioactive natural product, this discovery is, for long. So as the whole world forever remember you for this discovery. In my career, in last 3 decades, I discovered more than 60 new bioactive natural products from microorganisms, plants, and other sources. So these always cherish my mind. Another point is I would like to discuss here. You may find a bioactive compounds in the laboratory. It is active against the wheat blast fungus or any plant disease or even human disease causing pathogen. But it does not give you a guarantee that it would be used as a drug or commercial agrochemicals, because in case of commercial product, there are lots of other studies needed to see whether there is any side effect or not, whether the activity is strong enough and comparable to the synthetic compounds or chemicals currently being used against the microorganisms. So it has been estimated that out of 10,000 new discovery of agrochemicals, you can find one commercially useful drug or medicine or commercially exploitable bioactive natural products. Thank you very much for good question. Anyone?
We synthesis vincristine or vinblastine for use as anti-cancer or like that.
Vincristine and vinblastine, scientists discovered these two compounds as anti-cancer drug several years ago. And as a natural product chemist, if you go through the literature, you can find the whole process published in scientific articles, how they discovered those compounds. So those compounds, by following that already published protocols, you can easily get from the Noyantara or Catharanthus rogeus plant. So if you get discover these two compounds again from the Noyantara, it is not a discovery, because it is already been known, the analytical process is known, structure elucidation process is known. So it is not meaningful to discover again, and you cannot publish the data. But what you can do by following that technique, you can you know, uh, isolate and purify those compounds in large scale from Noyantara, and you can do the business because these compounds are very expensive. So by following the established protocol or known protocols and methods, you can you know, purify the compound in large scale. And if you have, you know, 1 gram of Nayantara or you know, Nayantara bean crystalline on bean plastic, you can be a very rich man. So obviously you have to understand that the amount of the compounds present in Nayantara is very low. So you need huge amount of plants to extract and get the sufficient amount of the anti-cancer drugs. Any more question? Do anyone have any more question? You can ask question because in your exam, these analytical methods are very important. You may be asked in your exam, suppose you have discovered a bacterium from the soil of BSMRU field, which is antagonistic to wheat blast fungus. And if you are in that case, how can you discover the fungicidal compound from the bacteria? I showed you how can you discover compound from the plant materials. In case of bacterium, suppose you found a bacterium, strain name is BTL 01, which is antagonistic. Antagonistic means, you know, inhibit the growth of wheat blast fungus. It means that this bacterium produces certain antibiotic which is inhibiting the wheat blast fungus. In that case, what you need to do, you need to culture the bacterium in liquid medium, for example, nutrient broth medium in a large scale, for example, 10 liters or like that. And after culturing the bacteria, you know, within a week in the flask, become turbid and huge billions of cells of bacteria will be produced if you put them in the nutrient medium. And then bacteria also produces, bacterial cells produce the antibiotic in the culture medium. So what you can do by using ethyl acetate, you can extract the compound secreted, produced and secreted by the bacteria in the medium. So by using ethyl acetate is water insoluble organic solvent. So if you add ethyl acetate equal volume and shake the flask, and then you will get ethyl acetate layer and water layer. Water layer would be in the down and ethyl acetate layer in the top, and you can separate the ethyl acetate layer. And by using vacuum you know, evaporator, obviously in every step you have to test the biological activity, that is its activity against the wheat blast is present or not. And when you do the fractionation, which fraction contains the you know, fungicidal principle, and then you go for the structure elucidation. So you may be asked like that way, or you may be asked, suppose you found a sea cucumber in the Saint Martin Island, which extract show antifungal activity against a fish disease. How can you discover the bioactive natural product present in that extract? So you have to, uh, show in a flow chart how the step-by-step process you follow to discover the compound. After the discovery and purification of the compound, you must do the bioassay to confirm that this is the pure compound and this is the bona fide bioactive compounds that I targeted from the extract. And sometimes, not a single compound, sometimes, 2 or 3 compounds may be responsible for the activity of the extract. So in that case, you say we discovered these 3 compounds or 2 compounds which are responsible for the anti, fungal activity against the wheat blast or against other organisms or like that. So any other question?
Assalamu alaikum. Sir, this is Adur.
Yes, other.
Sir, my question is, sir, different chromatographic method like TLC, HPLC, or gas chromatography. My question is, what method will be easier for us, or what will be the best?
This is a good question. How can you decide which, chromatographic method you use? Usually, when you get the extract, we use the TLC, thin layer chromatography, because it requires only micro liters of the extract, and it gives you the information, detailed information about the nature of the compounds present in your crude extract, the polarity, as well as you can use several different kinds of solvent picture like ethyl acetate, chloroform, or hex and hexane in different ratio. And then you can get the information, and TLC will give you the information what kind of solvent mixture you need to use in the column chromatography. So first you do the TLC, and even preparative TLC, if you find the spots are separated well from the preparative TLC, that means from scraping the silica gel from the TLC plate, you can elute the compound in each spot. You can get a very tiny amount of compound and check where is the biological activity, which one is the active. And if you get the good information, then you can precisely go for further chromatographic technique, whether HPLC or you go for the silica gel chromatography. Usually we use silica gel chromatography after TLC to get several fractions, and then we do the bioassay, because we need enough amount of compound. In case of column chromatography in the laboratory analysis, it will give us enough amount of compounds, because column chromatography you can do in a big column, and you can get milligram level purified compound. You must understand that to elucidate the structure, we need milligram, minimum 1 milligram, 100% or 99.9% purified compound. Otherwise, you cannot elucidate the structure. And until and unless you elucidate the structure, you cannot make a conclusion that which natural product you discovered. Any more question? Good.
Sir, assalamu alaikum, sir.
Yes.
Sabrina, sir.
Yes, Sabrina.
Sir, if we don't get enough amount of product from our extraction process, so is there any further process to increase the concentration of our product or increase the amount of our product?
Yeah, this is another good question. Yes, you don't know which compound is active in the crude extract. So in that case, after
Yes, sir.
all the process, and if you finally get very small elucidation of the structure, for example, mass spectrometry, which gives you the chemical formula as well as molecular weight of the compound, you can do by using a microgram of the compound. But once you get the compound, bioactive compound, then what you can do, you can repeat the same process using new extracts. So you need more, the source materials, for example, microorganism or plant materials, and do the same job. Once a protocol is developed, then no need to do any trial and error. You can easily go through the same protocol, use the same protocol and get more amount. So you need to repeat the experiment with more amount of the source materials to increase the amount. So usually, in case of plant materials, I used, minimum 1 kilogram of dried materials to grind and make them powder and then extracted with organic solvents. You must remember organic solvents, and if you have notebook, you can record. Polarity of the solvent is very important. Polarity means which organic solvent is high polar, which organic solvent is low polar. Polarity means high polar means the tendency to dissolve in water. That is, high polar compound, they are hydrophilic, and low polar compound hydrophobic. In organic solvent, which we use in the extraction as well as chromatography, they are, for example, N-hexane, diethyl ether, chloroform, ethyl acetate, acetone, and methanol. That means N-hexane is the uh, hydrophobic compound, and methanol is a hydrophilic compound. So the polarity of methanol is the highest, and polarity of N-hexane is the lowest. So you may be asked which compound is the higher polar compared to 2 or 3 or 4 compounds, or like that, or highest polar compound. So I am telling you again, N-hexane, diethyl ether, dichloromethane, chloroform, ethyl acetate, acetone, ethanol, and methanol. So ethanol, methanol, they are the bottom, and N-hexane is the top. N-hexane is the most hydrophobic, organic solvents. So oily compounds, they are dissolved in those solvents. You in chemistry, organic chemistry, you must remember another saying that like dissolves like. Like dissolves like means like dissolves like. That means organic high polar compound dissolve in high polar solvent. A low polar compound dissolve in low polar solvent. For example, if you discover a terpenoid compound which is oily, you cannot dissolve easily in ethyl acetate, water, or ethanol. In that case, as the compound is highly hydrophobic, oily, you can easily dissolve in N-hexane, or chloroform, or dichloromethane, or diethyl ether. So in your practical experience, in your household experience, you can see oil, any oil, cooking oil. If you put in water, you can see they are floating and they never dissolve, because oil is organic compound, and oil is hydrophobic compound. If you put oil in kerosene or organic solvent like chloroform, or ethyl acetate, or N-hexane, easily you can dissolve. So but salt, sodium chloride, you can dissolve very easily in water, because sodium chloride is an inorganic compound. and water is inorganic compound. So water solvent can easily dissolve because they are like, both are like, say, organic solvent dissolve organic compounds very easily. This is why it is said like dissolve like. Higher polar compound dissolve higher polar materials. How higher polar compounds can be dissolved in higher polar solvent, and low polar compound dissolved in low polar solvent. This is the principle in organic chemistry. Thank you very much. Any more question? Do anyone have any more question? If no more question, then I shall close today's lecture. And I wish all of you stay safe and in sound health. And this is really a bad time we are passing all over the world due to the pandemic COVID-19. So please take care yourself, as well as be sincere about your lifestyle and food health.
আমরা স্যার অ্যালকালয়েড এবং টারপিনয়েডের শীটগুলো পেয়েছি, কিন্তু ইন্ট্রোডাক্টরি স্যার লেকচারটা স্যার শীটগুলো স্যার পাইনি স্যার।
I can upload today. I shall upload you the introductory lecture today.
Okay sir, thank you sir.
But introductory lecture I gave you in the first lecture which I delivered at BSMRU campus. You didn't record that one? Then I shall give you. No problem. Any other question? If no question, then thank you very much.